home who spare their valuable time to be with us today this evening, and also all the scholars and the students who have come and the delegates for to attend this program. Once more, I would like to add, we welcome again Dr. Karthik, and we are associated like what Professor Dutta said, and I will give now all the time to him to give his talk on this day. Thank you.
Thank you. It's a great honor and pleasure to be here. I was an alumni of Utkal University, and that's what brings me back to this place. And it's a great occasion for me to connect with all my teachers, my friends, and colleagues who have worked with me during my PhD. So I really cherish this moment, and now connect with all the young students who are coming through this university. So today I'm going to talk about a project that I've been engaged with since I moved to CCMB, because CCMB is a CSIR institute, and it has a vast array of instruments which can be used to understand better organismal biology. And I took up work on how the polyvalent antivenom, which is very widely used for treatment of snake bites in India, can be improved. So the problem of snake bites in India is one of the neglected tropical diseases. I noticed that Professor Dash in the morning talked about neglected tropical diseases, but didn't mention the snake bites. And this is a very important neglected tropical disease. And there are many deaths that happen in India for various reasons, which I will go through in the next few minutes. So 65,000 people die in India. This is the recorded deaths from hospital data. And certainly this is an underestimate. We all know many people die without even going to the hospital. About 14 to 25 lakh bites happen in India every year. That causes the 65,000 deaths. And these bites are inflicted to people who work in the field. They're usually rural, poor people who don't have proper footwear or work involving hard work involving labor in the fields. So they cannot access readily medical help. And when this happens, it is very cruel to the family, because the person who's a breadwinner almost loses the life sometimes, or becomes completely morbid. There is loss of limb, or there is a condition of morbidity that they will not be able to earn for the family anymore. So it is a serious concern, considering the rural agrarian economy is facing a crisis because of many reasons, and this is adding to the burden that the agrarian population faces. So we are known as a land of snake shamans, right? But we really do not have the wherewithal to master the treatment of snake bite. We have more than 300 species of snakes in India, and only 60 are known to be venomous. Most of the fear for snakes is because of the venomous snakes, but they form a very small fraction of all the snakes that we have in this region. And it is almost inevitable, even if you walk across the road this evening, you might see a snake. So anybody in this country will encounter a snake during their lifetime. So it's not that people are removed from this problem. So I have just tried to prepare a mental map of this complex problem, and this requires a lot of effort which has to come through multiple disciplines. And the reason is the mortality is one, very high. There are very strong cultural beliefs, which means that snakes have some association with our mythology, and we consider them sacred. Sometimes we consider them vicious, and we hurt them. And this also creates problems in people accessing treatment and also treating snakes, properly in their environment. And then there is a strong reason to believe that many bites result in permanent physical disabilities. Additionally, because of the large ecology of various species, the niche of various species is pretty large. There is geographic variation in the venom. The treatment cost is very high, primarily because of the number of vials of antivenom that are required. Because of this reason, the efficacy of the polyvalent is very low. So typically, a treatment starts with a dosage of 10 vials and it can go 5 doses for a bite, which means 50 vials. One vial of antivenom costs 600 rupees in the market. So it is 6,000 per dose into 5, that is 30,000. Then typically a person bitten by a snake is admitted into the ICU because you need incubation, you need a heart-lung machine, everything ready. So they essentially brought into intensive care. So additionally, those costs built in, and it almost breaks down a low-income family. The person is bitten by a snake. Then there is delayed medical help because only district hospitals have such intensive care units. In the remote areas, you don't have facilities. And the same reason that I mentioned before, the geographic variation in the venoms result in large deficiencies in the polyvalent, which cannot be bridged at this point in time, unless we have regionally representative antivenoms. So the majority of the problem is caused by the big four. These 4 species are called the big four, and 2 of them are elapids and 2 of them are viperids. And elapids have a combination of both neurotoxic and chemotoxic venom. Viperids have majority chemotoxic venom. And in my lab, we work on 1 colubrid and 1 viperid, 1 elapid and 1 viperid. The elapid is the common trait, and the viperid is the phosphate viperid. So this is the a pictogram of how antivenom is produced. This is more than a 100-year-old method. This was started because the British wanted to occupy and live in India for long periods of time, which have lots of snakes. And the first thing they did was to produce antivenom. And when they did it, they used horses, which were used in their services when they were bringing their army, moving their army. There were many old horses. So the horses were brought in to use as an intermediary organism where they will be immunized by the venom, and blood is drawn from them, and then there is a step of antibody purification, and then they are lyophilized and ready for administration to any victim who might be bitten by the snake. So this process leads to many problems, which I will go through in the next few slides. So one concern is the animal welfare, and at every stage there are testing of animals involved, and which includes both costs as well as animal welfare concerns. And finally, the product that we get is not as efficacious as it should be. So I just divide the problem into 3 major groups. One is the effectiveness, the other is the affordability, and the third is accessibility and availability of data about the disease. And I will go through each one of them during my talk. So in terms of accessibility or availability of the antivenom, there is very poor understanding of the burden of snake bites in the first place in many parts of India. There is poor access, we know that. And how do we bridge it? So this is a survey that we are doing in one district, because most of the papers that we have, data which is being accumulated, is from district hospitals. And as I told you, many people don't even reach those hospitals. They either succumb to the bite or they go for remedies which are not conventional, or they do not require the polyvalent. They go to a painkiller and they get treatment, and they probably die after a few days, which is never recorded. So we took this district, district called Jagatia in Telangana. It's a newly carved state in South India, and we are now covering all the 320 villages in about 45 mandalas in this district. So, as all of you know, state is the public health is a state subject. The federal government doesn't have much say in what the state does with respect to the healthcare of the people. They can only give some recommendations and support. So eventually, the management of healthcare of people has to be done at the state level. So we wanted to generate this information at a very fine level, almost map every village and every outcome. All the greens are people who survived, their deaths and even morbidities, disabilities. So this has never been shown so far. People have talked about deaths, but disabilities is almost 1/3 of all the outcomes, which is very significant. And it needs to be factored because it includes livelihoods of people. So we just outlined the various factors that might lead to poor and environmental outcomes, which are related to accessibility or availability of polyvalent antivenom, which could be physical barriers, social barriers, economic barriers, and barriers resulting from lack of awareness. So we are building an epidemiological data at a district level, which can be eventually scaled up to the state. The second aspect is effectiveness, and this requires a lot of previous experimentation, because the venom is a cocktail and it varies from one region to another. So the first peek we had about this variation is when we looked at the soft-scale venom, soft-scale viper venom from Tamil Nadu, Goa, and Rajasthan. So three distinct ecoregions. And the venom from Tamil Nadu is often sourced to manufacture the polyvalent antivenom. So if you take this as a representative venom that is used to immunize the horse and derive the antibodies, you can see the variation that is happening from one ecoregion to another. It is usually the representation of different toxins. The proportion of certain toxin keeps varying from one region to another. And this is typically because the ecoregion has certain food types which are accessible to that snake. So we went on to find out how much of this is retained by the antivenom. And we looked at the retained and non-retained profiles, basically using an affinity chromatography. And we conjugated the antibodies with the beads here in this column, and then passed the venom through it. And then we have a control profile which shows the venom. These are the toxin peaks, and these are the non-retained peaks. Still, you have the low molecular toxins such as disintegrants and disdomain P2SDMPs, which are eluting out. That means they have no antibodies in the polyvalent. And this is the retained profile. Fortunately, many of the high molecular weight toxins have sufficient antibodies represented in the polyvalent antivenom. So again, the two regions, Tamil Nadu and Goa, had very different profiles of the immunoretained toxins. That means if a person is bitten in Goa and uses the polyvalent antivenom which is generated from venom which is used from Tamil Nadu, the efficacy of the antivenom reduces. And then when you talk of crates, there are 8 species of crates which are medically important in the country, but all the antivenom is manufactured from one crate, venom of one crate, which is the common crate. And these crates are distributed, 8 species are distributed all over India, and people are exposed to bites of these non-spectrum crates. So what is the outcome if a person is bitten by, say, Bacillus fasciatus? What will happen? So if you look at the profiles of 3 species, this is the you can call it the control profile or the profile of the species from which we generate the antivenom from Tamil Nadu. And Bacillus niger and Bacillus bungaroides, they are very distinct from Bacillus ceruleus. So if a person is bitten by bungaroides or niger, the antivenom which is manufactured using the venom from ceruleus will obviously not yield very positive results. Then the third aspect of affordability is also matched with what I just talked about in terms of efficiency. It is the number of vials that we require to utilize the venom toxins when it is in circulation inside the victim. And how do we improve the quality of the venom, polyvalent antivenom? So we performed several antivenomics experiments, and we tested geographically distinct snake venoms through antivenomic experiments that involved using site exclusion, affinity chromatography, where we put the venom, pass through the venom through beads which are conjugated with the antibodies, and we elute the non-retained fraction, and then we find out how much of the polyvalent complexes are formed for any particular venom. So we get what is called as a complex which is derived without the polyvalent and with the polyvalent. So you have 3 different, one with the venom, one with the antibodies, one with the conjugate, the complexes which are formed between the venom and the polyvalent. So when we do an overlap, you will find that the shift has happened already in the peaks because of the complexes being formed between the polyvalent and the venom. Polyvalent and venom and the venom. So you will find 3 peaks instead of 2 peaks as we found with the just the antivenom and the venom. The third peak which is resulting here is the complex. So we made an overlap the null spectral signature and the complex, and we found out what would be the area of the curve to match it with the efficacy of the antivenom. So when we plot it with different concentrations of the venom that we use against a known quantity of polyvalent, we can draw this area under the curve. So you get a Kmax through this, and this is the EC50 value. So when we arrive at a value like the area, the rate at which the binding happens between the polyvalent antivenom and the specific venom, you get different values for the 3 regions, and this can be used to check the efficiency of the venom for different efficiency of the PAV for different venoms. Now, it is not just the binding ability of the polyvalent. There are functional toxins that work on specific functions in the body, in the physiology of the human being or the host, which is being utilized here by the snake. There are consumption of coagulopathies, cardiovascular shock, then hemorrhage, extensive bleeding, internal damage to tissues, and then myotoxicity. All of this are caused by different toxins which are found in the cocktail of the venom. So we developed some functional assays, and we derived the same estimates of EC50 using these functional assays. One is the proteolytic assay, and the other is the PLA2 assay, and the third one is the clotting assay. Then all of them result in different values of EC50, which we finally summarize for the three regions. And obviously, we found that the average effectivity score for Tamil Nadu is the highest. That's because the venom is obtained from here to develop the polyvalent antivenom, and it is least for Rajasthan. And it is obviously a very distinctive ecoregion considering the source of the polyvalent antivenom is from the venom that is sourced from Tamil Nadu. So one hurdle that we face currently is the standardization of in vitro assays for neurotoxic venom. We are still doing this work, and we are using cell cultures, primarily neuronal cell lines, to look at how EC50 can be calculated by panning the cells with the great venoms. Now, after we calculate the ED50 and LD50, we can develop a correlation between the two, and we can actually replace all the animal experiments with in vitro assays. Now, this has two advantages. One is we are addressing the concern of use of animals in these experiments, extensive use of animals. Every batch is screened with several mice, and we can save all those lives of those mice. Additionally, we can also reduce the cost. All of this involves cost. And the values that we generate using the in vitro assays are probably far more accurate, and the use of the venom is also reduced. So all this results in better outcomes for the antivenom production. So some future perspectives I thought I'll introduce, considering that the audience comprises of several young people, because these are things that can be done in the future. Already there is evidence for this, that there can be venom gland organoids that can be made. There is already several groups in Europe who have developed organoids from venom glands. So there will be a perpetual source of a distinct snake venom through the venom gland organoids. We don't need to access the snakes every time. And it will allow a large amount of experimentation to be done on the variation of the venom through this development. The second is also developing large data on transcriptome, genome, and target genes which are expressed in the venom gland, to understand those which are toxin genes and non-toxin genes, and then go on to recombinant technologies which can be used to produce toxin-specific antibodies. And further, you have also the human parts display now available. So you can develop humanized antibodies for the snake venom. So just to summarize, we have all the three major gaps that we initially identified, and clearly the need for epidemiological data arises as a significant drawback for accessibility, improving accessibility. For effectiveness, the toxin composition is poorly understood. The techniques that are required to produce toxin-specific antibodies are still not there, and I highlighted some of the ways by which we could do that. And how do we address the problem of non-spectrum venoms, which I introduced in the problem that we face with the cranes. There are 8 species of cranes, and we have venom from only one of them. And affordability. There are many ways to reduce the cost of a single dose of antivenom. One of which is implementing 3 Rs and reducing the cost by introducing in vitro assays. And these have to be vetted by a lot of experiments which have to be done on animals initially, but eventually they can be completely replaced. And there are many approaches to producing more effective antivenom antibodies for snake venom, and all of which should be deployed. So thank you. And this is the team which is doing all the work. Siddharth was my first PhD student, and he worked on soft scale vipers. And this is Srinimas, who's all the time collecting data on snake bites in Jagatya district. This is Alka Sahu, and she works on trade venoms. And this is Apni, who's doing the whole genome of Echis carinatus soft scale viper. Thank you.
Thank you, Dr. Karthik. Now, I think many people would like to ask some questions. So, anyone who wants to.
Need extra cost. Suppose some small thing
it doesn't go through, it's not a contact poison. It's not, that's why they're called venoms, because they are injected inside. They have, the snakes have a specific apparatus.
small crack.
Yeah, it's possible. So we take all the precautions. Usually it's in a lyophilized form. We don't keep it in liquid form. And whenever required, small amount is taken and made in solution. And then very low dilution, very small quantity of the venom is used in micrograms.
That's why I'm asking this one. Since we are working on snake, everybody will be very frank. And then, so there is much data on molecular things or so that I will be awesome.
No, the bigger challenge is to extract venom. Yeah, so all my students are trained in snake handling and they have to extract venom. So that is more challenging than handling venom in the lab. Because we would be very happy if there is a solution to not use snakes anymore to extract venom if we develop organoids of the venom gland. So then we don't need to catch them. No, the reason Tamil Nadu is the source is because of the tribal group called Irula. And they were originally snake catchers who were catching snakes, and they were involved in the leather trade involving the snakes. And the snakes were brought into the Wildlife Protection Act. They were listed as endangered species and they should not be killed. And therefore, they were brought into the whole system by asking them to bring the snakes, and they would milk the snakes of the venom and release it back. So this started to rehabilitate them from an industry which was exploitative. So, but it has its problems. As I told you, it is not as effective in other regions.
Very nice presentation. So only I have a few queries. Right now, whatever the anti-venom are produced, that is based on their toxin profile or full venom.
The anti-venom, which is now, it's a polyvalent anti-venom. It's not monovalent. It's polyvalent. And it is by injecting the whole venom into the horse.
Yeah. That I understand, but you told that there is a geographical variations
Yeah.
among the different types of venom from the same species. Is it due to only their food habit or some, I think, some environmental influence or some epigenetic mechanism may be there because within the species when such a variation is there?
Yes, yes, very true. I think you have spotted the problem accurately. It is not purely genetic.
Yeah.
Okay, the genetic distance may not be correlated with the toxin difference that you find. These modifications are even post-translational.
Exactly.
Okay, there are some packaging that is happening in the venom gland which could result in the toxin.
So if we will go over the proteome mapping of venom versus toxin across the different group of the same species, that can give a better solution of developing better antivenom, I think.
Yeah, and just give you a small peek into some new information or already available information. Between males and females of the same species of snake, there's a big difference. So that also means that in a population itself, within a region, there is variation. And there are ontogenic shifts in the venom composition. From birth to, because snakes, as they are born, they have to hunt. So they also seek prey which are easily accessible based on the size. And then as they grow, they shift their diet. So,
Adaptive physiological response.
exactly. Yeah.
Thank you. Thank you.
Yeah.
Any more questions? All right.
The polyvalent antivenom is for the both for like toxic and nephrotoxic.
Yes.
Some snake bite someone like nephrotoxic. We don't know what snake bite. 100% effective.
Yeah, that's a good question. As I told you, it's polyvalent. So, it's a boon for a clinician because a clinician gets a person almost half dead. And that person says, don't die in my bed. I want you to survive. So they start putting the polyvalent antivenom without thinking what species of snake has bitten. Though the clinicians look at symptoms, for example, ptosis or muscle tonus, lack of muscle tonus, and respiratory dysfunction. All those things are indicator of muscle paralysis, which is more neurotoxic. It's the great venom that causes it. So if you have local damage, like tissue damage, then it is a viper venom. Okay, so clinicians look for symptoms, but they have ready polyvalent which can be immediately used when they know that it is a person who has been bitten by a venomous snake. So in a way, it is a boon for the clinicians, but there are problems because of the inefficacy of the polyvalent antivenom to bind with all the toxins. I gave you the example of the Saustier viper, where disintegrants and disdomain SBMPs were not bound by the polyvalent. They were just flowing through the pollen. So they will be in circulation and causing more and more damage to the victim. Though you might inject many volumes of the polyvalent antivenom, right? So it has something to do with the host organism like the horse that we are using. So the people are trying to now use camels because camel antibodies are small, and people are using alpaca, many other camelids now are coming in the picture. People are using goats for antibody generation. So the different iterations have been done to improve the efficacy of the polymer.
I have seen like in real that a person bitten by a common carrot.
Common?
Common carrot. Carrot.
Correct.
Sorry.
Correct.
Common carrot and another person is bitten by a cobra. Polyvalent antivenom used, but the common grade bitten person got some kidney disease, after some years. But in cobra, when one was okay, it's like 100% cured.
Yeah, so every bite is unique in the sense snakes don't bite to kill humans. This is a very wrong concept. They bite in defense. And the venoms are not made for defense. These are venoms to kill prey. So the venom toxicity is very high. In fact, a bite of a venomous snake like a cobra can kill 50 elephants. But it still injects that because it's not a defensive venom. It is a killing. It's to kill the prey. So but it doesn't do it to kill the human. It is actually using it to defend. So therefore, it is known that the snakes meet the venom. They don't want to use the venom unnecessarily. So sometimes the bites are dry. So the cobra bite that you are referring to could be a dry. Okay, so you cannot infer based on the outcome that the antivenom has actually worked. And in the case of the great bite, you mentioned about a kidney problem. This is becoming quite apparent in the eastern part of India, where acute kidney disorder is coming to be a very important long-term morbidity of people who have suffered snake bites, particularly viperines. I am first time hearing things.
I have
Yeah.
To avoid certain behaviors or reduce the risk of being bitten? And can you instruct physicians, for instance, to refer more quickly in case of suspected bites or to change the treatment so that the outcomes could improve, or are they doing the best they can?
Yeah, I think this is going to help. Every death you can avoid is going to be good. So yes, behavioral change, like for example, using boots and not walking in the night without a light. Simple behavioral changes can reduce the extent of envenomations in people. But I would not think it would reduce the deaths. Because the deaths are caused because of other factors, like physical barriers. There's no access road to even the incident happens way inside somewhere, and to reach the road and reach the district hospital it takes 5 hours or 6 hours. So that you cannot change.
So it's crystal. You could airlift someone. I mean, in the helicopters. Next, but maybe this is not an Indian.
Yeah, it's difficult in a developing country like that. So access to medication is a major issue. But yes, behavioral change will certainly avoid many bites are avoided.
My question is, I used to ask this question. I have asked times in different forums. Is it possible that one snake will bite another?
Yeah.
And if that happens, is it that the other snake will die?
Yes. 100%.
Okay. People
Yeah.
used to say no, they are not, they are
No,
not.
there's no immunity to snake bites. Yeah.
Okay.
Whether the snake will identify
No, I was telling you about snake.
I have faced a similar occasion when I was not seeing on the road. Morning, I took photograph in the summer March in Coimbatore, in Mardamai Hills now. I came and cobra was it seems, and then without seeing this one, I was walking, and then one cobra was in front of me. When I was standing in front of my nose, like that, then I step back 2 steps, Then he went away. Then again I stepped 2 steps further. Again it came like that. Then I ran. Then he went whether male or female. But as a scientist working on snake, whether it has any behavioral mechanism to identify human or other animals in wild, something like that. It is a common question because I am a human being, so that I am asking this. Okay. Yeah. Yes.
No, I think they do recognize threat. It could be a dog, it could be a human being. So they also want to remain safe. So they don't want to die in vain. So they will threaten you to, or that's why it raises the hood and it doesn't bite. So it gives many chances. Then it raises the hood, it charges, then it bites. So it gives so many chances for the person to retrieve.
So it appears that snake attack / bite to human is completely defensive for defensive purposes. So in that case, then do they want to conserve as much as venom and not inject venom in many cases? Is that the theory?
That's what I was saying. They seem to show venom metering. That is, they will deliver venom only the quantity that is required. They have that ability to control with the muscles and the venom glands and also the action of the. It's basically a hypodermic syringe kind of mechanism. So when it goes in, it's a high pressure insertion into the skin, and then there is a negative pressure going inside the fat, and then they equilibrate it and inject the venom back into the system. So they can control it.
Sure.
So there is some muscular.
So that's at least comforting that they don't want to upload the whole venom on a human being. I mean, for me, when I saw a picture there, last picture, one student handling, I'm afraid of even throw a snake. And the young lady kind of maneuvering snake, that's awesome. I salute your students for doing what they are doing. Thank you.
Thank you so much, Dr. Karthik Vasudevan, for giving us this speech, and also for us to be aware about the most problem that are facing by especially people in the rural areas, the farmers, and taking up this aspect to bring also about an awareness to the people in the rural areas, especially how to protect themselves. Like what if they go at night, they should not walk badly. What to protect themselves with a torchlight or something like that. So thank you so much, and we're looking forward to have in the dinner time for more questions want to be asked by any of our members who have attended this program.
May I request Professor Rupa Guru Madam to felicitate our speaker, Dr. Karthikeyan Vasudevan. Madam, please.
Her achievements are: she is the invited speaker for National University of Educational Planning and Administration's International Conference on Governance Issues in University 2017. ICML appointed her a chair and invited speaker in Asia's Symposium on
Yeah, sure. Don't know what the time is because my phone ran out of battery, but it's 8. All of us are kind of looking forward to the end of the sessions. So, very briefly, I would like to touch upon the areas that my lab works on. In fact, I'm more in the mood of sharing anecdotes rather than hard data because I don't think our brains can process it after that heavy lunch and the end of the day, and a lot of very interesting and diverse things that our brains, our collective brains have had to process. I think we will briefly touch upon certain areas that may be interesting because the theme of the seminar? National Conference on Innovations in Animal Science Research and Social Transformation. So we are zoologists here. I do not know if there are people from any other subjects, maybe life sciences, and yes, microbiology, I met somebody. So it's an umbrella where animals are used along with humans, because I know the anthropology department is very powerful here. It's a spectrum of living forms that move and that we use as repositories of knowledge using various technological extrapolations, which is rather an arrogant term for humans. Nonetheless, we are here to discuss the various extrapolations that could be made in the various systems that the animal world offers to us to study, to understand, to empathize, and to coexist. So I have asked one of my students to prepare the slides for me because I wasn't even aware of the amount of time that should be allotted to me. By the time the program reached me, I had already boarded the train. So there will be many slides, but Payal has, my scholar, has very thoughtfully put in those bullets that Professor Gunanidhi Sahu had sent me, which brings me to the part where I thank the organizers, Madam Robhati and Professor Gunanidhi and Dr. Subhangshu and Bikash, who has also recently joined the department, and Professor Bandapat, who I met. So I hope we shall have some time for interactions. So the talk mainly touches upon little thematic areas that this conference is very thoughtfully put together. And the sort of the objective of any conference is to allow for networking among students, amongst young investigators, and amongst established investigators in various branches of science. And I do hope that happens. So, role of biological science in social transformation. I wouldn't go so far as to say that social transformation is the end objective of any scientist or technologist, because science and technology subjects are sort of taught in a very Western, with all due respect, way in which there's a lot of arrogance and entitlement bred in the minds of the students who are studying science and technology. So it's not like we are here to make any kind of social transformation, but I guess it happens anyway. So there are a lot of mechanisms put into place whereby certain ethical paradigms are kept in mind, are taught to the students, and I would like to stress upon, for example, in the talk preceding mine, somebody was saying catching the snakes. So capacity building is a very important part of any kind of animal research, and people who directly work with animals will appreciate that properly handling of the animals probably give them the message that
this is not to harm us. And of course, I'm not talking about the sacrificial experiments. Many of our drug discovery preclinical studies are end-stage experiments, but generally correctly handling the animals is part of what is taught in drug discovery preclinical studies. So this role of biological science in social transformation aspect, a bit of translational research may be introduced at this point. Many of the talks from the morning, one on anti-helminths, one on integrated crop management, studying the biology of different animals, I think malaria control and dengue control. So translational research is basically something that you apply directly in terms of processes or products that can benefit the humankind as well as environment. Outcomes research aims to understand the end results of particular healthcare practices and interventions, which also includes studying the toxicity, the pharmacology of various compounds that are harvested, sometimes out of plants, sometimes out of animals, out of microbes, or synthesized, taking cues from what is available naturally. At this point, we probably, even if we don't want, our funding agencies and our ethical committees, administrators, policymakers, and society in our country, a lot of the research is funded by public money, makes us aware that science and community must work hand in hand in the generation of knowledge and wealth, and the science and technology must percolate through academics, through industry, in the highly skilled manpower that are generated by academia, have to be trained to work for the knowledge industry, and policy that works on the seamless marriage between academia and industry and society and community, which brings us to various kinds of bench to bedside studies that should be that are made, for want of a better word, customized to serve certain needs of society, which could be in terms of value of care, in terms of quality improvement, in terms of real-world effectiveness and quality measurement of various services that are provided for human health care in the pharma sector, in the biotech sector that works directly with outcome of treatments given to humans, and for environment, because ultimately it is the health of the environment that ensures or not our collective survival. So translational research in my lab with my collaborators who are people who work in the field, normal people, I mean regular people who may not be associated with academics or scientific labs. The media plays a very important role. So what we do is we connect the dots, and seminars like this are also platforms to connect the dots. Only if the stakeholders are many in number and in types do real world solutions come out of such platforms. So you can see the cold is getting a little allergy, and we work on allergy. So this is a very, very real situation. I don't know whether the positive hormones are being secreted in your brains right now, and if you're feeling very energetic, even if you're not, just take solace in the fact that we do what we do, but ultimately we change the systems irrevocably, and the system autocorrects. So connecting of the dots is simply leveraging those connections in order to what we feel is a positive end. So everybody sort of come together, the general public who fund the research. Right now, we need to make the public aware, especially in government-funded jobs, of what we are doing in our labs. So the media plays a very important role. And as we go through the lecture, maybe some of this will be discussed in more detail. So advanced techniques in biological sciences that are used in our lab include drug discovery that use natural products. And like I said, I have a big team and we use various bioactive compounds which have been characterized and are available in the pure form, purified form, or extracts that we take directly from the plant sources. So the one like I said, I will make my lecture a little anecdotal. So we will go to pan. I know in Odisha people are fond of pan, as in Bengal, and it has recreational values. It is also used in Ayurveda. It is also used in various rituals. However, often it is misunderstood in the sense that certain pan cultivars, even though they are genetically same, cause uh, gum cancer and or inflammation in the gum. However, our studies show that not all pans are similar. Some pans do yield very powerful antioxidant, anti-inflammatory, and pro-regenerative. So degeneration is irrevocable loss of tissue that is reversed. So the study here showed, and when we were applying for a grant, the study section said, oh, lots of studies have been done on pan. But the nuance here is, according to the cultivars, according to the regions where they are cultivated, there is difference in the medicinal chemistry profile of the plant, and with the difference in the bioactive compounds, the physiological effect is also different. So these are little things that come out of it. With chalkumro, because I do not know what it is called in Korea, chalkumra is like a whiteboard. And during COVID, when we didn't know when we could go out in the market and buy our next vegetables, in Bengal, we often also fry the skin of the vegetable, which normally we throw away, and we fry it with posto. What is the English of posto, Pradipta? Poppy seeds. And it's a very delightful dish, but often the skin is thrown away because there are studies on pesticides accumulating in the skin. So during COVID, when our maids were not coming and we didn't know when we could shop for our next food stuff, and we were conserving our food, so we were using the skin for eating too. And one of my students, the student in fact who has prepared this slide, is working on the water extract of the skin of chalkumro on chronic kidney disease. And that very day, she shared data. We were all working online at that time, and she shared data that has shown excellent results in both acute kidney injury and chronic kidney disease. So these are little, anecdotal things that we do. Drug discovery using natural products. So various phytochemicals. We consciously look for molecules, or we sometimes work with collaborators. Sometimes we work because a funding agency has a thematic area, and whether by chance or by design, we come upon various compounds which this slide will show you. And for example, phycetine, which is available in berries, is used in various diseases.
No, ma'am, just excuse me, there's a small announcement. Just 1 minute. Take poster presentation will start right now. So those who are about to present their posters, they are requested to be there near their posters.
So then, who do I talk to if most of the people are going to be gone? Okay, I guess I can talk to the chairs or to the speaker who is obliged to wait till I finish.
I
Okay.
think only the participants who will be responsible for the poster, the rest can be here.
Okay. Okay, so these are used for various diseases such as asthma and idiopathic pulmonary disease, idiopathic pulmonary fibrosis, peritonitis, atopic dermatitis. So A, you can have the fruits directly as functional food. B, you could have the bioactive compounds in a formulation. Guar gum is the other a natural product which is nanotized and it is used. Hard it is used in rheumatoid arthritis, an extract of it. And so these are some of the other stories. So these are some of the publications. The translational outcomes are we are generating information on bioactive compounds, on food that could be had, local food, seasonal food, which often get bypassed, and or formulations that could come out of the bioactive compounds, which may normally not be getting assimilated in the system, because only polar compounds get directly assimilated when we take in the food. In regenerative medicine, which is the other aspect of what my team works on, we work on tissue engineering of stem cells of various sources, embryonic stem cells, adult-derived stem cells, placental stem cells, and we use them for various purposes. We also use a planaria, which is a diploblastic animal, as a model, where we use its system to understand the process of regeneration and wound healing. In idiopathic pulmonary fibrosis, which is quite common in urban setups, stem cell therapy has been recommended to renew and rejuvenate tissues which are dying or are dead. So somehow we should reverse the process. I think during Professor Roth's talk, somebody was asking about reversing the aging process. So there are stem cells in all adult tissues which normally do it in old or disease systems when they are perishing. Stem cells are there, and somehow we need to augment the turnover of the stem cells to achieve a desired goal. So what you see in the slide, so this is the human embryonic stem cells, and these are human umbilical cord derived mesenchymal stem cells. These are bone marrow derived mesenchymal stem cells. And the various growth factors are studied, secretions and secretomes from other cells are studied, so accessory cells that normally work in the tissue. Hypoxic and hyperoxic conditions are studied. So basically, we're trying to study the various landscape or cellscape, for a better word, that can nudge very gently the cells towards the desired developmental outcome in a disease and on old person in which the turnover has been very, very less. So these are some data from the mice, and since there are students, these are various techniques that are taught to the students that they use for the drug discovery purposes. In pharma companies and or biotech companies, zoology students are of very high demand because this is the only subject that teaches the architecture of various animals that may be used for the purpose. So, quickly passing over, these are cells in a xenobiotic transplantation model. We have tissue engineered human embryonic stem cell line into alveolar epithelial type 1 and 2, which are the cells of your lung, which you lose gradually as the lung starts losing its function. We have used various cells. So the earlier data was on a cell line, and this is on another. This is of Indian origin. So the other was funded by an R01 grant, and this one is funded by DBT. So the tissue engineering basically plays with the cellscape to understand what best nudges it towards a desired outcome without directly tampering with the genes. So these are translational outcomes that we are working with. We work with the kidney and the cell for regeneration. We work with probiotics, which are friendly microbes that we combine to understand whether the secretomes could be of any use to us. This particular one was used in a preclinical disease model of IBD, inflammatory bowel disease, and we saw excellent results which make us wonder if we could repurpose the drugs for other purposes, such as the gut-brain axis, which also determines the wellness, the mental wellness of an individual. So these are some of the data out of it. CFU is colony forming units assay, where we measure the biogenic potential of stem cells. So the stem cells only grow in particular media, the cells die. And so in a tissue which is functional, we can measure the number of stem cells that there are. In degenerative tissues, they go down in number. So these are probably how the probiotics work. They either compete with the bad guys which come in our system and protect them in the form of a biofilm, or change cell-to-cell junctional anomalies which tend to make the cells leaky. So this is how they probably work. So this is a product that we have, and some of my students have startups, and they work to take them to the market. This is some of the mechanisms by which they work, and this is one of our probiotics. Okay. Planaria, since this is animal science, planaria, I told you, it's a very small animal, does not need ethical clearance. Like I said, it's very arrogant. Some animals need ethical clearance because they have eyes and they can scream in pain, and others who don't need ethical clearance. So these are immortal animals, and you could cut them into 3 pieces, and each piece will grow into a full animal. It's like a hydra. So we use it for understanding certain regenerative outcomes in a cell biological platform. So this is how they look like. So the other aspect that we work on, biotechnology, nanoscience, and bioinformatics, I think Professor Karthikeyan before me was talking about antibodies. We had a brief discussion in the morning. So versus fossils, we use camels. We humanize the camels, but we engineer the rest of the process in an E. coli system. So it's a huge 300 kilogram animal, and we take about 3 ml of blood from its popliteal vein, and that's it. So the antibodies that we have raised are against certain pro-inflammatory enzymes, and this is a proof of concept. However, these can be used for various purposes, such as diagnostics, therapy, anti-venom, as we learned about now. So any antigen you give, we have the patented technology to generate a large number of clones from which we can generate the antibody proteins and purify it. So this is basically the process that is used. In this case, we have used IgE from mouse, which has been challenged with allergen. So this is basically, it's a very involved process, but it's pretty simple and can be done in 4 to 5 days. And since the antibodies are very small, they are not thermolabile, which is the problem with most monoclonal antibodies. You have to store them in a cold temperature, and if the cold chain is broken, they perish. This is not a problem with them. So this is what some of my students have done, various antibodies that we've generated. And well, like I said, this is, the media plays a very important role in bringing this to the notice of the public with whose money we work. We also work with nanomaterials. Boer is one that I mentioned. We use it for various purposes, including probes. So if you have a drug that you want to see where it goes and what it does, you tag it with a probe, which you can trace. So this is one. This is an assay that shows uptake over time. This is a galactose mannose competition for uptake by the poor nano surface functionalized probe that we have generated. These are some of the assays to validate low cytotoxicity. This is a model of thioglycolate-induced peritonitis, which is basically in any kind of surgery. When your body cavity is violated, you're likely to have septicemia. So this is an aseptic model where instead of LPS, we have used thioglycolate that generates the exact same molecular mechanism and migration of inflammatory cells that must be controlled if the the patient is not to die immediately of septic shock. So this is also something that we have worked with poor nano with. Okay, so these are atopic dermatitis. Atopic dermatitis has no cure, only steroids are administered. So this is a natural product that has shown commendable results. We validate it through various kinds of cell-based assays, various kinds of cells, their growing techniques. So these are also part of the capacity building that animal sciences labs offer. So this offers a 3D matrix for cell scaffold for any kind of regenerative surgery. Biodiversity conservation and economic sustainability. So like I said, she has touched upon many of the points that the conference wanted to highlight. So at the end, we are a 106-year-old department, and we have a museum, which I'm sure all zoology students will be aware of. And so restoration of the museum, when I first came, and 30 years before I joined, the museum's last employee had retired and the university did not replace him. So many of the samples didn't have tags on them. And they were in a state of decomposition because nobody had bothered to change the preservatives over time. So then I chanced upon DNA barcoding to identify. And since we are a drug discovery lab and a technology development lab, there was a lot of media furor at one time that there were a lot of spiders in Medinipur in West Bengal, which was attacking humans. So then a local councillor requested me if you could identify it. And so we started working on spiders, and they turned out to be tarantulas, which are a very primitive form of animals, and they were not attacking humans. Rather, because we were destroying their habitats, in May and June, they looked for mates. So the poor guys were looking for females, and anybody going out at night were getting into conflict. So this is the work that we do. We are interested in the technological part. These are some of the books. This is the museum that I thought zoology department would be a good place to share the anecdotes with. I saw a very nice diorama opposite to the head's office. So this is the repository, the www.zoologyhub.org that you can go to. And so this is basically what we do: natural products, cell-based therapy, and nanoparticles. And yeah, knowledge and money is important. Various applications and repurposing of existing and or novel drugs through biological assays, tissue engineering of cells to derive products, cells, or processes that could be used, providing drink and additive. Right now we are into, like I said, we have startups that work on these things, nanomaterials and nanoscaffolds. We also work on these animals to understand who they are here in the museum. And those two are our model animals, aside from the mice. And so this is what we do, and these are my students, and these are my funding agencies, and thank you very much.
All right, thank you, Dr. Manandi, for the lecture that you have given. Now I would request for any us would like to ask any questions.
Thank you, madam, for your nice presentation. So I would like to ask one question that what is the name of the cell that causes the regeneration in planaria? And if that cell will be,
Neoblast.
if that will be interrupted to any other animal, whether the regeneration power will increase?
That's a good question. We haven't really tried, but the neoblasts won't survive in another animal unless we also explant a bit of the stroma with it, because it will need somewhere to grow. So if it is completely foreign territory, it is likely to perish, which is true for any kind of graft. Even if we do autologous graft in our own bodies, if the stroma is not right, where we explant it or implant it, it will not survive. But if we assure that, mostly with immunosuppression in vertebrates, but I don't know in invertebrates, maybe it will, maybe it won't. But okay, it's an interesting experiment, and since I don't have to go through ethical clearance, maybe I will take your suggestion and see what happens.
Any more questions?
Many people to ask questions, and or anyway they are tired. But thank you for the opportunity, and thank you for your hospitality, Utkal University Department of Zoology.
Professor Banerjee, thank you so much for the lecture within a very short span. The laboratory that you're working on natural products, drug discovery, and regenerative medicine, which is one of the important focus in this. So thank you so much, and wish you all the best and also.
Now may I request Professor Guru to felicitate the participant.
Now, I would like to invite on stage Dr. P. Gaurishankar, sir, for his talk. Dr. P. Gaurishankar is a wildlife biologist and TED speaker. His interest lay in evolutionary biology and in studying the phylogeography of king cobras across Southeast Asia. He has received the PhD from Maharaja Sri Ramchandra Bhanjadeo University, Barivada, Odisha, India. He is also a former exchange student at Uppsala University, Sweden. He has been awarded the Herpetologist of the Year 2015 by the Swedish Herpetological Society. Sir is the co-founder of Director of Kalinga Foundation. Sir has worked at premier organizations like Center of Herpetology or Madras Crocodile Bank Trust. Dr. P. Gaurishankar has helped set up the Augenville Rainforest Research Station, Augenville, Karnataka. He has been featured in several wildlife documentaries like King Cobra and I, Secrets of the King Cobra, 1 Million Snake Bites, Asia's Deadliest Snakes, and King Cobra, by channels like the BBC, Discovery, Nat Geo Wild, Animal Planet, Smithsonian, and National Geographic Channel. His title of the talk is Phylogenetic Study of the King Cobras, that is Ophiophagus henna, with new insights on classification, naming, distribution, range, and conservation. So,
Thank you. I'm going to finish really quick because most of the introduction methodology, what I wanted to talk about, most of them I've finished it. So I can directly go into the result section, but I just came through the slide. So, but this is one thing I wanted to introduce because what species it is, species we're going to talk about, because we just heard Karthik talking about the venom, right? If you want to know the venom variation, first you want to know what species you're talking about. Whether these species, because they look alike or they look different, whether the species is the same species or different species. If you don't answer that fundamental question, then you won't be able to deal with the venom. If you don't deal with the venom, we won't be able to control 60,000 or 58,000 people dying every year. So that is why I thought I'll just talk about the species. A species is one individual or a group of individuals who can interbreed or breed and get offspring. So that is species. There's no way we can breed with chimpanzees. We were talking about the genetic variation. We might be close to the chimpanzees, apes, like 98% similarity, but there's 2% at least difference between them and us. So we won't be able to breed with them. So that is humans, Homo sapiens, we are different species. Chimpanzees are different species. Lions are different species. Okay? Of course, there are too many theories of species concepts. I'm not going to talk about that. Next. Oh. Sorry. That's one. Sorry. Okay. Oh. Okay. So even the computer is tired. Okay, so there are different ways of speciation. Look at that. Skip that. So they all look so similar, right? For example, look at that first slide. This is not working. The pointer. Okay, look at this. What do you see? A, B, and C. They all look so similar, right? Check that. There is slight variation between A, B, and C. So this is called cryptic species. So that means we can't even think about venom variation when the species itself not solved. That is what I was trying to do. Okay, so this is important. I wanted to talk about the venomous snakes aspect, but I hope you remember what Karthik and mentioned. He has talked so much about the venomous problem that we have in India, so I'm going to skip that. Okay, now that's my study species, king cobra, one of the longest venomous snake in the world, and it can kill an elephant in 15-20 minutes or maybe 1 hour. It can kill me in 15 minutes. So that's the amount of venom we are talking about. So this species, for almost 185 years, look at that. I was talking about they look completely different from different regions, different habitat, and the species was described in 1836. Until 2014, up to now, it's just one species. Though there was speculation, but no one could establish that it's different species. That's my study site. That is where Karthik was talking about. He was talking mostly about India, but the entire Southeast Asia, we do have snake bite issue. So close to 50,000, 60,000 in India alone. So 150,000 people died all over the world. And India is the capital city for the snake bite deaths. So I was looking at how the species are so different, why they are different, and if they are different, when did they evolve? So I collected the morphology data and the tissues which includes bones, liver, whatever is shed skin. Yes, I have to capture a king cobra if it is a live one specimen to collect. I've got close to 350 king cobras, managed to collect close to 200 samples across from the zoos, captivity, like this, from museums and live specimens. So these are the characters, the morphological characters I considered, and this is the first drawing sketch of the description. If you see, that's 1836 by Cantor, and I used mitochondrial and nuclear genes. I don't need to talk about DNA to you people. So first results what I got was 4 clusters, right? If you see, so this itself gave me enough confidence that king cobra is not one species, there could be many. And I ran again something called as haplotype. All these are new softwares. I want to talk about the technology, how it's changed over the years, doing the DNA work that helped us to do molecular work. Otherwise, the old way, old school of comparing the different species was just the morphology part. Now, with the molecular work, it helped a lot. This is the difference I was talking about between chimpanzees and human beings. Similarly, I did this genetic p-distance. If you see a population 1 and population 2, there is 1% difference. Similarly, if you see population 3 and population 4, there is 1.6% difference. So the second gene, it's almost 5.45%. That means there are 2 different species or 3 different species. Morphologically, they look so different. Look at this. That's one species, second species, third one, fourth one. Physically, if I collect these samples across the world, that is the first species which has like 50 bands or less than 45 bands. This will have 70 bands. This has very, very light, more than 70 bands, and this was 0 bands. So that is how morphologically they look completely different. Look at this. These are juveniles. Why I'm talking about these things, connect again to Karthik's talk. It is very important to solve the problem. So unless you know the different species, you won't work. So my next future project will be to look at the venom variation of the species and then come up with anti-venom. For right now, as of now, there is no anti-venom for our Indian king cobra. When I say Indian king cobra, there's a anti-venom for this one and this one, not even this one. So for this is our Indian king cobra, so there is no anti-venom. So if somebody gets bitten, we will die. So this is quite important. And I've got bitten, and somehow managed to survive for various reasons. So this is quite important. So presently, like I said, it was one species for almost 185 years. Now it's 4 different species. Western Ghats in India, from Goa all the way to Tamil Nadu, is one population. The second population, right from northern Andhra, Orissa, Uttarakhand, all the way to Cambodia, including our Andaman Islands, it's second population. The third population, right here. The fourth, look at that. So that is the isolated population that could be a highly endangered species as of now. This is the one which Karthik already spoke. I'm not going to talk about that. This number keeps changing because, like I said, we don't have records. In some of the places in Orissa or in Sundarbans, there are islands where the people get bitten. I don't think so they even go to the hospital because there's no way they can reach the hospital. So this number changes. Karthik already spoke about these. Anyway, this is my team. As Dr. Datta and Gunanandi, you all know, Datta is my mentor and godfather. Karthik Shankar. That's him from IISc. I worked in his lab. Vijay, Priyanka, and Rihanna, they did all the lab work. And, yeah, that's Wolfgang with the snake. He's from Bangalore University. Indranil Das. So that's my. Thank you. Any question?
Now it's open for questions and
That is, snakes, king cobras eat other snakes, nothing else, exclusively on other snakes. It's called Ophiophagous hana. Ophio means snake, phagus means eating. That's the scientific name. Snake eating snake. Indian snake, Thailand snake, they all eat snakes. Not like us, vegetarians, non-vegetarians. Yes?
Any snake eat another snake or only that?
Correct. The crate also will eat other snakes. So all reptiles eat anything they can overpower, kill, and swallow. Anything can go into their mouth, they eat. A rat snake, a common rat snake, a non-venomous, that is without venom, can eat a Russell viper, which is highly venomous. A rat snake can eat a cobra can eat a Russell can eat a cobra.
Cannibalistic within their own cohort?
Cannibalistic? Yes, sir. King cobra is known for cannibalistic. Even cobras, all snakes eat. Even crocodiles, they eat other crocodiles, the young ones. They're cannibals. They're opportunistic, basically. Yes.
Sir, in your slide, you have shown it that there are some differences in the sequence, in the genome.
Yes.
And that is in few percentages, 0.54, and that in some points.
Yes.
So that is the whole genome comparison you have done.
No, it's not a whole genome. It's just a segment that is the 16S ND4. I chose 3 mitochondrial genes and 3 nuclear genes. So it's just a 400, 500 base pair sequences markers, not the whole genome.
Sir, is there any molecular mechanism which you have studied that is responsible for the structural differences for we are getting in different regions?
Yeah, I think, see, that's what I wanted to talk. The speciation happens when the two populations get isolated. When they stop breeding, then when they stop changing, exchanging, or there is gene flow stops, that is when a new species happens.
Sir, this is the evolution founder thing.
Correct.
Actually. But is there any molecular mechanism which you have studied? Means at the level of central dogma, at the level of transcription or translation?
No. I just use these genes to know the variation in their molecular, genes. That's it. Nothing much. And the morphology. You saw the band patterns, right?
Thank you so much.
Thank you.
Sir, is it homology modeling that you have done, that in silico homology? Have you taken up that?
Sorry, ma'am.
Bioinformatics tool you are using?
No, I used a few analysis like PPP, a few bioinformatics, but I don't know how the technology works. But I did use all the softwares, Yeah. Yes. Yes. Oh, yes, go ahead. Sorry, she's waiting for the mic.
So you use morphological and DNA differences to infer whether you're dealing with a different species. Fair enough. But as you said, the definition is a function. Can you get interbreeding, and will the offspring, if it occurs, be fertilized? So have that kind of experiments been done at all?
You mean the two populations can interbreed, you're asking? Yes, they can. If you put them in captivity, they do. And we've done that mistake before we got to know whether they do different populations. Yeah, they're fertile.
Then, by definition, they belong to the same species.
Yes, they can interbreed. They're not supposed to, but this was in captivity, not in wild. If you see the two populations, they can't interbreed. But which we did in captivity. Yeah, I wanted to show you the difference between two populations. There's no way they can interbreed as of given situation. There's a dry Deccan plateau which separates these two populations. But if you put them in captivity, they breed. Yeah. Maybe. Yes.
Sir, what will be the reason that Indian species don't have any antivenom?
because we just keen to conduct IPL matches and international matches, Bahubali movies. We make one of the highest expensive movies, and we are interested in those things. We don't mind people dying. Like I think there was the tropical, neglected tropical diseases. We have priority-wise there's so much. Actually, even the snake bite is one of the neglected tropical diseases. It's like 17th number. Actually, WHO removed this. But later, with a lot of pressure, with herpetologists across the world, they put pressure to the WHO, where they put back snake bite as one of the neglected diseases back. So we don't care when people die. So that's why we don't make it. Nobody wants it. Maybe people like me who handle them, work with them, 5 or 6 people, it's okay.
Sir, you are now in the coming years planning for do that antivenom.
Yes.
So what will be your plan for the future?
First thing I answered now. So now I'll be looking at the venom variation across the Western Ghats at least, and then do the antivenom test and everything, and we'll come up with the antivenom. But luckily, the king cobras in India, at least the Western Ghats, there are no bites happening. But in other Southeast Asia, it's quite serious issue. In Andaman, there are more king cobras than cobras. More people die due to king cobras than other snakes. So it is an issue. Thailand, they have their antivenom because their priority is different. But India, no, not yet.
Is it in endangered types of?
Yes, after my study, it will be endangered. Actually, Western Ghats will be endangered, and the Luzon population in Philippines, the small orange dot, that will be the highly endangered one. Yes. The IUCN status, everything will change after my study now. So my first paper is published, the second paper with the names is coming up. After that, I'm going to propose to the IUCN to change all the status of the species. It will change. Good, done. Thank you very much. Long day. Thank you very much. The organizers giving me a chance.
Thank you so much, Dr. Gauri, for the talk. And especially, I would like to mention, it's a very challenging work working with the snakes. So I think maybe this will motivate also some of our young researchers to take up the work which very few people are working now.
They will not take up because they'll die.
Thank you.
You don't know whether you will see me next year.
May I request now Professor Guru Madam to kindly felicitate Dr. Gauri Shankar. In fact, Dr. Gauri Shankar's notification of PhD degree was announced today evening only. So we should congratulate him. It's a really rare occasion that the very day of his award of PhD degree, we have the original water with us. And I still remember that the first day I met him, he showed us wonderful photos of king cobras. So I was really fascinated, and I was really happy to know that he will be joining us in this international conference. It's a wonderful one. Congratulations, and you have proved that molecular taxonomy is the taxonomy.
Yes.
That tells us only Western morphology taxonomy cannot be done. We have to depend on the molecular taxonomy. So, thank you. And may I now once again request Guru Madam to kindly felicitate Gauri Shankar.
This is the first presentation where I saw before my name there's doctor. A lot of times, because of my work and everything, people would announce that Dr. Gauri Shankar. I was like, no, I'm not done yet. But today I'm gladly or happily I can accept that Gauri Shankar doctor. Thank you, ma'am.
And also my heartfelt thanks to Professor Guru for her patience and chairing this session. That is the third session of today's meeting. Thank you, madam. And little announcement that because of some technical problems, we were supposed to have some cultural activities and our students. But unfortunately, it is not possible today. So let's hope we can have it tomorrow. And, so now for today, okay, the poster part is still continuing, pilot part, and the session that is the presentation is over for today. Let us meet tomorrow morning. So we'll start at 10 o'clock. So thank you very much. Yes, for your attention and patience, and I should not praise myself that it was a great success, but I was really excited in the morning that our Vice Chancellor also joined us soon after the meeting. During the presentation of Professor Walter, she came, but she told no, I did not disturb, and she was happy to see everyone, and particularly the students who went upstairs, and she was glad to see that students prefer sitting in the balcony and taking good photos. So hopefully we'll get some better photos from students. Sometimes it happens. So the photo which is best over here, we collected from the students. So thank you everybody, and I invite one and all present here for a dinner in the Department of Zoology. So I believe by 8 o'clock it will be over, and once again thank you very much.
Once again, I would like to thank all respected resource persons for coming again from abroad and from at home, and also all my scholars and students and all the delegates for your patience from early in the morning. We'll finish for dinner. We have a very hectic day, I think, but it was successful. And I've seen that it was very diversified, really the